process, I became an expert in isolating mucus-like material. And they knew this at Rochester, and one day, one of the professors came by my lab, Alex Downs. He was a chemist who had worked at Cornell on crystallizing proteins. And he was interested in cell nuclei. I had a fancy method of isolating the nuclei of cells. And he came by one day and said, "Look, Simmons, see this test tube?" He stirred around with a glass rod. And then he said, "Now, look, if I add a little ammonia to this, it's going to get so viscous that it's all gooey and stick to the thing." He said, "That's got to be the DNA. Because nothing else would be that gooey in the nucleus of a cell." He said, you're good at isolating all this. Do you think you could isolate DNA?" And I said, "Well, I'll try. It sounds interesting." I was getting a little bored with what I was doing. So I set out to do it. And I looked up what there was known about in the literature. There was the only procedure that had been published was one which would be impossible to isolate DNA from. You took a solution of this viscous of nuclei which you collected, add a high concentration of salt, it would separate the thing, become all gooey, and then you'd have to remove the protein little by a laborious shaking with chloroform and that sort of thing, centrifuging, and you got rid of 10% every time you did it. So it took you 10 manipulations to get rid of 90% of the protein. Meanwhile, all the enzymes were attacking the DNA, and it couldn't possibly survive that. When one day, my good friend and colleague, Alejandro Zaffaroni, who was a postdoctoral fellow there, came by with an article that he'd seen in Nature showing that a large dye, a big dye, orange G, which is a sulfated dye, a huge dye, would attach to proteins. The acidic dye would attach to the basic part of the protein, form it to precipitate. You could wash that precipitate and then dissociate it again, make it go back into solution by the addition of sodium dodecyl sulfate. Sodium dodecyl sulfate is a long chain fatty acid, a sulfated acid. Instead of a carboxyl group, it would make it a soap. It's a sulfate group, and that binds to the protein and dislodges the big orange dye, which then goes into solution. Then you can see measure the concentration of the protein by how much was precipitated by the dye. Orange dye is released and goes into solution and gives you a colored solution, and you can measure the protein by the color of the solution, depending on how intense the color is. Well, I said, that's exactly what I need to know. I want to know how to to release the DNA from the protein and precipitate the protein and let the protein, it's marvelous, which was wonderful. It accomplished everything except that particular compound could form my cells, and it was difficult to work with. It was a soapy kind of a thing. And I found something else, sodium xylene sulfate, which could do the same thing and wouldn't form these soapy solutions. And in one swell food, you could take a material which had nuclei, you could add the sodium xylene sulfonate, that would release the DNA and precipitate all the protein quantitatively in one step. How it go? Which was exactly what I wanted. But we had a dividend which I didn't realize about until many years later. That not only did it separate the DNA from the protein, but it destroyed all the attacking enzymes that would attack the DNA in the process. There's nothing else that could have done that. And so here, not only was I able to isolate the protein the DNA completely free of protein, and at one time it was protected from being attacked by all the attacking enzymes, which permitted me to isolate DNA for the first time that was pure, free of all protein, and undegraded, unattacked in any way. And I gave that paper at the American Chemical Society in New York in 1952 on the isolation of the 10-hour preparation of DNA, the isolation of pure, undegraded DNA. One was from Paul Doty at Harvard, who had one of his graduate students there because Paul Doty was interested in the physical structure of DNA. And another was from Maurice Wilkins in London, who was an X-ray crystallographer, who was studying the structure of DNA by X-ray diffraction methods. And Rosalind Franklin was working in his laboratory, either as a graduate student or as a postdoctoral fellow, I can't remember. And she's the one that did all the work. He did the X-ray diffraction analysis. Now, another incredibly serendipitous event occurred which made the elucidation of the structure of the DNA possible. In order to make sure that I had pure DNA, I subjected it to a variety of tests. Analytical ultracentrifuge to see that it was one molecule size. Electrophoresis, which would tell you that it was a pure compound with an electric charge that would migrate without any other contaminants as a single peak. And in order to see if the carbon, hydrogen, nitrogen, phosphate would correspond to what the theoretical composition was of pure DNA, I had to send samples out for analysis. What I did was to dry the sample down, to get a dry weight basis. I put this in a Soxhlet extractor, which heats the thing in vacuum up to 110 degrees, drives off the water, and I sent that out, and we never got the right answer. It was always, everything was always too low. It occurred to me that there was water bound to the DNA, and by heating it, I was breaking down the DNA and binding the water permanently to it so that there was always a certain amount of water bound that could not be removed and would give the rest of the analyses too low a figure. So I decided to not heat it, but to dry it in vacuum over P2O5, phosphorus pentoxide, which picks up water very strongly, until it had could lose no more weight at room temperature in vacuum. In other words, dry it out without heating it. I was going to send that to the analyst for the elemental analyses when it occurred to me that it's going to pick up water. And if his laboratory is humid, it's going to pick up moisture. So I needed to know exactly how much moisture was picked up by the DNA according to what the ambient humidity was in the laboratory. Now there are solutions which you can make which in a closed container will give the air above it humidities of 20%, 40%, 60%, 80%, etc. And I did that. I took little samples of DNA, put it in at humidity of 20, 40, 60, 80% humidity, measured the weight gain, and I could see how much water was absorbed onto the DNA molecule at 20%, 40, 60, 80% humidity, ambient humidity. And it was approximately the same. At 20% humidity, it picked up 20% water, 40, etc. 60%, 80%, 80% humidity was 80% water. When I sent the samples, I equilibrated the samples at 40% humidity, which was my laboratory, which would be his laboratory humidity also, so that that was not going to change, and I could tell him it had 40% moisture, which he could subtract from his weight, and he, for the first time, he got the correct carbon, hydrogen, nitrogen, phosphorus analyses. He had been the analyst for the Rockefeller Institute in New York, and he had done the DNA, and of course never got the right answers for anything. And when he sent me back the results, he put a big congratulations in red crayon all over the whole sheet. He was just as excited as I. When I sent the sample to Morris Wilkins, I said it contains that moisture. And he wrote back some time ago, no, he said it can't, because it seems to me like it contains more like 80% moisture. And I wrote back to him, of course, that the reason it has 80% moisture is because your lab in London has 80% humidity. And that little observation was what made the analysis of DNA possible. Because they had been doing all their X-ray diffraction analyses in vacuum, in which they dried the molecule down, free of all water. And when they looked at it, the reflections in the X-ray looked like a pack of stacked discs. You could not see any other structure, except that there were reflections that looked like stacked discs. When I told him about how it bound water as a function of the ambient humidity, Rosalind Franklin, he decided, well, instead of drying it down, it would equilibrate at 80% moisture, and then do the X-ray diffraction analysis and see what they got. Well, of course, when they put it at 80% humidity, the molecule expanded just like one of those slinky toys, which when it's flat looks like stacked discs, and when it's expanded you can see as a helix. When they did the analyses at 80% humidity, there they could see that indeed it was a helix. And it was Jim Watson and Francis Crick, who were at Cambridge at the time, saw those diffraction guys because they knew Rosalind Franklin, and they set to figure out how this could form a helix and give all the parameters that were known about it, that it would be degraded by temperature, etc., which meant it was a hydrogen bonded structure. And the two of them together, Francis Crick was a crystallographer, Jim Watson was a clever guy, and the two of them came up with the hydrogen bonded, double-stranded structure of DNA, which meant, of course, that this was the mirror image, the two mirror images of each other, double-stranded. If you took them each molecule apart, you would have the mirror image of another, and this meant that this was a possible way of reproducing, and they offered the hypothesis that genetic replication was possible by virtue of the fact that you could separate the two strands and you could form identical pairs by forming a new strand where the old strand was on both